And where do I even start with Marta? So some of you are from outside of the community, or you're within the EV community. And I think, broadly speaking, we know Marta as this highly cooperative person, passionately committed to community, right? Like she single-handedly started a Bloomington co-op by buying a house and refurbishing it. She, using her Halloween costume, she is now the face of a strong political protest, and you know what I mean, if you know what I mean. She organized a conference for scientists in the Midwest, broadly, or co-organized it. And I think all of those sort of, in their own absurd way, capture Marta's generous dedication to community. And I think that permeates in her work. All of her work, first authored and not, is highly collaborative with grad students and undergrads alike. So another thing to note is that Marta's highly dedicated to travel, sometimes during the middle of those cooperative experiments. And some of her travels brought her early on to the Kellogg Biological Station. And that is where I would say she learned a core way of thinking that permeates all of her work. So she learned how to program at a field station and using maximum likelihood and Bayesian statistics that I think forms the glue of her work. And also produced one of our favorite stories of her naked bike race, which she's enthusiastically told her committee members who didn't necessarily want to know about it. I have to share that I'm kind of surprised that her work has so much field data permeated throughout it, because I kind of thought that the university would ban her from using our vehicles anymore after she crashed a truck really on in her work, well, severely damaged a truck. Nonetheless, every chapter has tons of field data within it. And maybe this one just caps it all, right? So despite crashing the truck, they still let her drive an F-150 equipped with a Mustang engine. And returning back from field work one time, she, riding with Alex as co-pilot, wailed on the gas to pass someone, screaming, F-150, beep! Sending a cooler with samples flying everywhere on 2nd Street at 37, which you know is a place if you get out of the car, you can die. Yet somehow, Marta and Alex collected all of the samples, none of which broke, which I think is just symbolic. Like there's just some way for Marta that things just work out. Well, we'll see if they work out in a warmer world with disease. Marta, thermoecology of zooplankton, take it away.
I just need to collect myself for a second.
There's so much more. I mean, we could just.
Okay. Thank you, everybody, for coming. Thank you, Spencer, for that introduction. Okay. So there was a lot of concern starting in the late 1990s over how climate change and global warming would affect the spread of infectious diseases. So here's an iconic map from that time period showing how malaria risk is projected to change by 2020, only 4 years away. You'll notice Indiana here, dark orange. This map is pretty scary. There's not a lot of blue, a lot of other colors. But in the few years after this, there was sort of a backlash to this. And I think it's sort of epitomized by this quote from our favorite surfer ecologist, Kevin Lafferty, who said that the projected global increase in infectious diseases with climate change suggests a pending societal crisis. The result has been the emergence of a crisis discipline reminiscent of the early phases of conservation biology. So this is not a compliment, right? He's saying that basically we're letting our hysteria drive our science. Our assumptions are sort of leading us astray and can lead us to the wrong conclusions. And so a decade and a half later, we still don't really know how temperatures affect the spread of disease, even not in the context of climate change. There's a lot of unknowns still out there. And so how do we answer this question without giving into the crisis discipline mindset? And so I think the answer to this, and I think Kevin would agree with me, is that we need to let the science lead us, and we need to be really careful about determining causality. We need to be really rigorous in looking at lots of different kinds of mechanisms, and we need to connect evidence across different scales. We need to look at field observations, lab experiments, and use models, and sort of the interplay between them to tell us how temperatures are affecting the spread of infectious diseases. And sort of the foundation of that is mechanistic or process-based models for temperature and disease. So what do I mean by this? I mean, we'll take temperature, look at how that affects specific host and parasite traits that we know are really important for regulating the spread of disease, and then find some way to synthesize them into a measure of disease spread. And so there are a couple different measures of disease spread. One of the most common ones is called R0, and I'll be talking about that later. Another related one is called transmission potential, but they work basically the same way. As they get bigger, you have a better spreading disease. So if we look at a quantity like R0, we have infection gains, things that lead to more disease in the numerator, and then things that lead to infection losses in the denominator. So I'm going to walk you through an example for malaria, which I think is a disease that most people are at least a little familiar with. The typical equation used for R0 in malaria has number of vectors in the numerator, right? The more mosquitoes that there are, the more malaria spread you'll have. The faster or more frequently those mosquitoes bite, the more disease spread you'll have. The better those mosquitoes are at transmitting disease. And then in the denominator, we have things like vector death rate. So if mosquitoes are dying really quickly before the parasite can develop inside them, we might expect less disease spread. So we can sort of fill out this equation and use it as a way to synthesize how all of these traits are going to impact the spread of disease. But what do these traits actually look like? So if we have temperature on the x-axis and a generic trait value on the y-axis, we can do an experiment and measure that trait across a gradient of temperatures, get some data, and then we can fit a function that best describes how that trait depends on temperature. And so this is really what we're combining. Seems pretty simple, but there's actually a lot of uncertainty. One of the things that leads to uncertainty is just the fact that this shape is kind of a weird shape. It's sort of exponentially increasing and then turns over really fast. And so you have to be really careful and precise about how you characterize this downturn, or you might get the wrong answer. And that's actually the story behind that first map that I showed you. And why we don't think that that's actually going to be true is that it didn't correctly incorporate this downturn for some of those traits. And I'll talk about that towards the end of my talk. Another thing is that there can be tension between traits that have opposing patterns on disease spread. So again, going to malaria as an example, if we have parasite development rate increasing with temperature, so when it's hot, parasites develop really quickly. When it's cold, it takes a long time. But host lifespan decreasing. This one, we would expect temperature to increase disease spread. For this trait, we would expect temperature to decrease disease spread. So how do we know where the peak actually is? And so that's where this synthetic approach comes in handy. So the case study I'm going to talk to you today is a zooplankton-fungus disease system. And I'm going to explain to you why warmer is sicker in the system, because transmission rate increases really strongly with disease. Not only that, but there is a trans-host effect of past temperature. And so past temperature, the temperature that spores are made, is vital for understanding how temperature regulates this disease. And that high temperatures are probably not going to constrain the spread of this disease as far as we can tell. So here's our host. It's a crustacean. It's about the size of a comma. They're very cute. And so here we have 2 uninfected hosts. They're really clear. You can see the heart here in particular is basically translucent. And then here we have 2 infected hosts, and they're just chock full of spores. The heart here is just a giant dark ball of doom. And so a lot of the traits, like the rate at which these hosts become infected, the rate at which spores grow, are strongly impacted by temperature. And so these little ladies, live in lakes across the Midwestern United States, including Indiana, where our field sites are. And so if you've ever been swimming outdoors, you might guess that the lake water temperature varies quite a bit over the course of the epidemic season. So diving into the lake and taking a closer look at how the disease is transmitted, we have susceptible hosts hanging out in the water column, feeding on algae. And while they're feeding on algae, they'll also be incidentally consuming fungal spores that are just hanging out in the water column. And they do so at a foraging rate F that's temperature dependent. They'll become infected at a transmission rate beta that's also temperature dependent. And then when infected hosts die, they release spores into the water at a spore yield rate that we call sigma. So our typical seasonal epidemics start in late summer or early fall and wane in early winter. And as I mentioned, while this is happening, the water temperature is steadily decreasing from the summer maximum. So I'm going to start by giving you 2 field patterns that we need to solve with our toolkit. So the first problem is warmer sicker, and if so, why? So here are some examples of 4 different epidemics from the same year in 4 different lakes. And you can see that some epidemics start really early and get huge, like up to 50 or 60%. And some epidemics start later and they're just pretty wimpy. So it seems like temperature could be driving the size of epidemics. And we can actually rigorously quantify this relationship by integrating the area underneath those epidemic time series curves. So now I have temperature on the x-axis and epidemic size on the y-axis. So each of these points is a single epidemic in a single lake in a single year. And there's a pretty strong relationship. There's lots of noise, but still a very strong relationship between the temperature when an epidemic starts and how big it ultimately becomes. So is this causal? And if so, why? The second field pattern that we're going to look at is, can it get too hot for disease? So if we look at the temperature when epidemics start here in this dark gray graph, you can see they start at a variety of temperatures, as you might guess from the previous pattern, but that most of them are below the summer maximum temperature, and that no epidemics start when the top layer of water is above 30 degrees. So it's suggestive that maybe hot temperatures are constraining disease. Maybe we've hit that steep downturn on our thermal reaction arm. Okay, so now that we have some observations of natural populations to show our relevance, we can combine that with individual level experiments and parameterized models to give us a good idea of what mechanisms might be driving it, and to resolve the tension between different traits. And then test these predictions from the models with population level experiments to really nail down causality. And it's really the interplay between these three approaches that I think would satisfy Kevin in us being able to really show that temperature is increasing the spread of disease. So the outline for today, first I'm going to talk about why warmer is sicker, and I'm going to talk about it in two parts. First, I'm going to use the R0 whole transmission process synthetic approach to show you that transmission rate is really what's driving this pattern. So we'll measure a bunch of traits and synthesize them into a measure of disease spread. And then we'll test that with a population level mesocosm experiment. In the second part of this first part, I'm going to show you that a trans-host effect of past temperature is actually really critical for understanding how temperature regulates disease in the system. And so we'll build another mechanistic model. We'll test that with some experiments using field-collected spores. And then we'll do some model simulations to show how that might matter for epidemic seasonality. And then finally, we'll move on to the question of, can it get too hot? And the answer is probably not. And we'll be using a very similar approach to the one we used up here, but at higher temperatures. And throughout all of this, I want to keep returning to this theme of that we're resolving tension between different traits that are impacting disease spread in different ways. Okay, so first part of the first part. So outline for this section, first we're going to get an equation for R0 as a function of host and parasite traits. We're going to measure those traits across a temperature gradient, combine the traits to get a function for overall disease spread, and then test it with the mesocosm. So to get our equation for R0, I'm going to return to this original schematic that I showed you. And so we've already identified most of the important traits. Foraging rate is going to be really important. Transmission rate is going to be really important. And spore yield is going to be really important. There's two other traits that we need to know. The first one is the density of susceptible hosts, which you can think of as like fuel for the epidemic fire, right? That's why we vaccinate, because we're trying to get rid of susceptible people in the population. And so this star, that just means that it's a conglomeration of other traits that I'm not going to show you. The other important thing is going to be algae, which again is a conglomeration of other traits. And the reason why algae is so important is because this spore yield trait doesn't just depend on temperature, it also depends on the food resources that the hosts are eating. So if they eat more food or better food, they'll produce more spores. The energy just gets passed on directly to the parasite, basically. Okay, so here's our equation for R0, and I'll walk you through it. So remember, things in the numerator help spread disease, things in the denominator help inhibit disease. So spore yield is in the numerator, unsurprisingly, more spores, more disease. And then you'll see that spore yield is also a function of algae. This H here just makes it a saturating function, so it doesn't keep on increasing forever. At some point, you just can't eat any more algae. Transmission rate, more if you become infected at a higher rate, more disease. And then more susceptible hosts, more disease. In the numerator, we have spore loss, which does not depend on temperature. Host foraging rate, because hosts are actually removing spores from the environment as they eat them. And then that's weighted by the number of hosts, since they're doing the removing. Okay, so foraging rate increases as a function of temperature over the entire relevant temperature range that our epidemics start in. Spore yield has a unimodal shape, so we are getting this downturn in the spore yield trait. Transmission rate also increases over the entire temperature range. Algae is increasing, and susceptible hosts are decreasing. So when we plug these into our equation, we can sort of think about how we might expect R naught to change. So spore yield is hump shaped, so we might expect disease spread to peak at a middle temperature. Algae, we would expect to increase disease spread with temperature. Transmission rate, we would also expect to increase disease with temperature. But foraging rate and density of susceptible hosts, we would expect to decrease disease with temperature. So again, we have this tension. What's going to happen when we put all of these things together? And the answer is pretty clear. Disease spread increases with temperature. So warmer is sicker in the system. And we can use the model to identify which traits are most important. And so it turns out it's transmission rate here and spore yield. And so if we look not just at the qualitative shape of these traits, but the magnitude of how they're changing, we can see that transmission rate is increasing almost 5-fold. So that's huge. Spore yield's only increasing by about 50%. So we think most of it is probably due to transmission rate, but this sort of downturn here is what's causing the leveling off at high temperatures. So now we want to test this result in a mesocosm experiment at the population level. So we had 33-liter fish tanks. We had 3 different temperatures. And so sort of just to plot those along our knot prediction, we're expecting just an increase in temperature of disease with temperature. And we let these experimental epidemics run for 56 days, which is about 3 to 5 infection cycles. Okay, so here's what the epidemic time series looks like. So we have time on the x-axis and infection prevalence on the y-axis, and you can see that it looks like epidemic size is increasing with temperature. And if we quantify this by integrating area underneath the epidemic curves, we see that it's definitely the case. And this pattern is actually really reminiscent of the field pattern that I showed you at the beginning. And so here, each point is a single tank. And we had 3 replicates at each temperature. So again, warmer is sicker. And we were actually also able to measure spore yield in infected animals from this experiment, and it did not change with temperature. So spore yield seems to be really highly variable and actually not that sensitive to temperature when you're not exquisitely controlling all of the other variables. So we infer that transmission rate is the primary cause. So conclusions for this part: warm temperatures promote disease spread in our system. They do so through changes in transmission rate, and that this contributes to variation in epidemic size that we can see in nature. Okay, so now I'm going to talk to you about the trans-host effect of past temperature. So the past environment can be really important for determining traits of various organisms. So for parasites, we call these trans-host effects, and it just means that conditions in the previous infection influence the pathogen performance in the subsequent infections. So you're probably more familiar with the concept of maternal effects, which is the more general term applied to most organisms. And I think the most familiar example is the Dutch hunger winter, which happened at the end of World War II, in which food supply was cut off for a very short amount of time, but very severely. And so children who were born to mothers who carried them at that time were much smaller. And actually their children ended up being much smaller as well. So we can see these environmental conditions perpetuating through generations in most organisms, or infections in parasites and pathogens. So how would this actually work with temperature in our system? So if we have spores that are contacting a host in what I'll call infection 1, that host becomes infected, we can harvest those spores, expose another host to them and infection 2, and then measure how the traits in this infection are affected by the environment here. So we'll call this our past rearing temperature, and this our current temperature. Okay, so the outline for this section, first we're going to build a model of transmission rate, and this time we're going to use a 2-step process for transmission rate, where first we look at how exposure changes due to foraging rate, and then everything else that's after that. And then we're going to vary past and current temperature. We'll parameterize it with experiments. We'll test our results with field-collected spores to show that trans-host effects do matter in nature, that spores reared at warm temperatures are much more infective than spores reared at cold temperatures. And we'll use model simulations to show how that could impact seasonality of epidemics. Okay, so here's our 2-step transmission model. So we have transmission rate here at the top, and we're dividing it into the product of 2 things. Foraging rate, which determines how many spores hosts come into contact with, and then what we'll call per spore infectivity, which is just everything else. So foraging rate depends just on current temperature and just depends on host physiology. Per spore infectivity, on the other hand, depends on both past and current temperature, and this past temperature effect depends only on this parasite physiology, the temperature that spores are reared at, while the current effect depends on both the parasite and the host and their interaction. And so we already have foraging rate as a function of temperature from the first part that I showed you. And so we can use that to determine how infectivity is changing as a function of past and current temperature. But in order to do that, we need to be able to manipulate past and current temperature independently, right? In order to separate out their effects. So we needed to do a factorial experiment where we vary them in combinations. So we measured transmission at 5 current temperatures and 4 past temperatures crossed for 20 total treatments. And then we calculated per spore infectivity as a function of past and current temperature by dividing this transmission rate by the foraging rate at the current temperature. And when we do that, here are the results. So it's a little intimidating, but I'll walk you through it. We have current temperature here on the front axis, and then past temperature going back. So each row in a color is one past temperature of spores, one batch of spore quality, you can think of it. And so we can fit a plane here, which is just a simple linear model of past and current temperature. And this plane describes how per spore infectivity, here on the y-axis, depends on current and past temperature. And so what these arrows here along the edge are showing you is that per spore infectivity actually decreases with past temperature, but it decreases with current temperature, but increases with past temperature. And past temperature is actually, the effect of past temperature is actually much stronger. So overall, what we find is that warm-reared spores are really good and infectious, and cold-reared spores are pretty crappy at causing infection. Yeah, so warm-reared spores are more effective, and the past temperature effect dominates. And so if we think about how this might play out in nature, we expect past and current temperature to actually be pretty tightly correlated, right? Because you're going to be infected by spores that were probably made somewhat recently. And so if we follow past and current temperature being equal to each other across this plane, we see that per spore infectivity should decrease over the epidemic season because the effective past temperature is larger. Okay, so now what happens when we scale this back up to transmission rate? Because transmission rate is what we're interested in. Transmission rate is what actually is influencing disease. So we can use our model to multiply back perp spore infectivity by foraging rate and see how we think transmission rate should change. And so here are the results. The model is the solid lines, and the actual data is in the dashed lines. And so the first thing you'll notice is the past temperature effect, right? So each of these lines in different colors is one batch of past reared spores. So as spores get reared at warmer and warmer temperatures, there's a general increase in transmission rate. But you'll also notice that the past rearing temperature of spores is really important for determining how transmission rate depends on current temperature. So when spores are really good and super infectious, when they're reared at warm temperatures, transmission rate increases with current temperature. And that's what we saw in the previous experiment. But if your spores are really crappy and not that infectious, there's actually either no relationship or almost a decreasing relationship with current temperature. So we have sort of the tension between past and current temperature at the level of per spore infectivity. And then we also have the tension between per spore infectivity and foraging rate. So past temperature is really modifying the impact of current temperature, so that transmission rate increases with current temperature for good spores, but not for crappy spores. And if you look at these points here that I have circled in black, those are the past and current temperature being equal. You get a much larger effect than you would varying past or current temperature alone. So the transhost effect of past temperature and the foraging effect of current temperature are really working together to make transmission rate super sensitive in response to temperature. Okay, so now we want to test, is this real? That's like a cool model, but it's kind of crazy, right? So can we actually measure this in the field? And we can. So I went out and I collected spores in -3 degree rainy weather. If I tipped over, I might have died. But we got the data, and that's what matters. And we showed that in 2 of 3 lakes, spore infectivity got significantly lower as temperature dropped in nature. And in this 3rd lake that wasn't significant, the spores just weren't all that good to begin with. So it seems like maybe there just wasn't that much room to decrease. Okay. So how might this impact seasonality of epidemics? Well, we can figure this out by simulating epidemics where we can pick a sort of imaginary temperature trajectory. So here we have temperature as a function of time. And then we can hold those transmission rates, so foraging rate and per spore infectivity, constant at this high temperature, or let them vary with temperature as it increases, and see how that changes the shape of epidemics. So if we hold all the traits or if we hold foraging rate and per spore infectivity constant, we see that epidemics peak and then don't really decrease that much at all. We'd expect full-blown 50% prevalence rolling into December. But if we add in the temperature dependence of per spore infectivity, so this is that transhost effect that's also moderated a bit by current temperature, we can see that now epidemics are actually starting to decrease quite a bit as it gets colder. If we add in foraging rate only, we can see it actually has an even stronger effect on epidemics. And if we incorporate them together, we actually start to see epidemics that look almost like what we observe in nature. So I think the temperature dependence of transmission could be, or is a good explanation for why epidemics look like they do in nature. So conclusions for this section: warmer is sicker because hosts are exposed to more spores due to their increased foraging rate. There's a trans-host effect on spore quality, where warmer spores are way more infectious. And both of these mechanisms contribute to the seasonality of epidemics. Okay, so the final section: can it get too hot? I don't think so. I'll show you why. So just returning to our motivating field pattern, remember I showed you that epidemics start at a variety of temperatures, but never above 30 degrees, and usually quite after lakes begin to cool. We can actually look at this in another way. Going back to this other field pattern, you can see this epidemic that sort of tries to start early in August, and then it's just like, it can't do it. It's like trying to take off, but it's not able to invade the host population. So maybe there's a thermal constraint. Maybe we've reached the downturn of the reaction norm, and so warmer is sicker up to a point, and then it's not anymore. So we can test this using the same strategy that we did before. And it turns out that a lot of hosts can actually withstand higher temperatures than their parasites. So you can imagine this as sort of mismatched thermal reaction norms, where the host has their downturn at a higher level than the parasite. So if we can get into this nice little zone of thermal exclusion, hot temperatures could constrain the spread of disease. And you're probably familiar with this concept already. Sometimes when you get sick, your body temperature elevates to try and diminish pathogen growth inside you. It turns out that ectotherms actually commonly have fevers as well. They're just behaviorally regulated, so they can use microhabitat selection to change their body temperature. And it's very well documented in the grasshopper, cricket, locust group. There's specifically a bunch of fungal pathogens that they can completely cure themselves of, given the appropriate environment or opportunity to bask and cook off the fungus. So fungi are pretty sensitive to temperature. Maybe that's happening in our system. Although we don't really think that the Daphnia are choosing to hang out in these hot temperatures, we know that they do it already. So Daphnia vertically migrate. It's just a part of their natural history. So they hang out in the bottom colder layers during the day to avoid fish predation primarily. And then at night, they come up to the top to take advantage of better algae near the surface, and they can grow faster in hotter temperatures. So maybe while they're migrating, they're cooking off the fungus. Okay, so in order to test this idea of a possible environmental fever, we need to, again, measure traits and see how they're affected by super high temperatures. So there's a few ways that this could work, right? So it could affect the foraging rate of hosts. And maybe hosts feed more slowly, so they're contacting fewer spores. So we measured it, and it doesn't seem like the case. There's a very maybe small decline here, but foraging rate at 30 degrees is still way higher than at 20 degrees. Okay, so that's not it. Well, maybe the hot temperatures are impacting the parasite's ability to cause infection once it's inside the host. So that's not the case either. Infectivity was actually remarkably constant, like not significantly different, like to 3 decimal places. I've never seen that before. What the heck? So if you look at transmission rate as a function of temperature, it just exactly mirrors how the response of foraging rate. Okay, well, what about spore yield? So if we look at spore yield, it actually does decrease at the highest temperature, but not very much. So if we combine these two things together into transmission potential, we can see that the effective foraging rate and transmission rate way overpowers the effect of spore yield. And so we expect more disease when it's really hot, than when it's not. Okay, so environmental fever is a bust. That's unfortunate because I thought it would be so cool. I would get like an amazing paper out of it. Well, maybe there's some other mechanisms that we could look at too. I told you in the last part that trans-host effects of the past-varying temperature can be really important. So maybe there's a trans-host effect here. So we looked at that, and there is, again, a trans-host effect. It's pretty weak, but let's add it in and see what happens. Okay, so now disease spread at super high temperatures is equal to 20 degrees. That's an improvement, but it still doesn't explain why we don't see epidemics at high temperatures. Okay, is there anything else? Well, one thing that we know is that high temperatures can decrease the survival of parasites in a free living stage in the environment. So maybe when the spores are just hanging out in the water column, they're negatively affected by temperatures before they even reach the host. So we measured that, and it turns out that after 7 days, infection or infectivity decreases by about 97%, which is pretty crazy. And if we add this in, then yeah, we get way, so this is on a log scale, we get way less infection at high temperatures than we do otherwise. But remember that I said this is after 7 days. And so we also did an incubation for 1 day, and there was no decline after just 24 hours. So we're actually currently redoing this experiment with finer time gradations to see at what time scale it might decline. I don't have this data yet. But we don't really think that spores are hanging out in the water column for that long. We think that based on sort of physical sinking and just the fact that there are so many hosts there eating the algae and the spores in the water column, that the spores are not hanging around that long. So this could matter, but we don't think so. The duration of these spores in the environment is something that's really hard to measure, so we're sort of thinking about how we might go about that, but it's not something that we have a good handle on yet. So conclusions for this part: hot temperatures are probably not a constraint on summer epidemics. There was no environmental fever for transmission rate and a very weak one for spore yield. The trans-host effect on infectivity helps out a little bit, but it's still not enough. And then the decay of free-living spores might help, but we don't really think so. Okay, so conclusions overall. Warmer is definitely sicker in our system. And the reason why that occurs is because transmission rate is super sensitive to temperature. And transmission rate increases due to the combined effects of foraging on host exposure to parasites, and the fact that warm-air spores are so much more infective. And then we think that hot temperatures are probably not a constraint. And there are some other things that might be constraining epidemics then, and you'll feel free to ask me about those afterwards. So take-home messages overall. Are we in a crisis discipline? Are hot temperatures going to solve everything? I think the pendulum has probably swung too far the other way. I think that we've gone from thinking like, oh no, oh god, we're all going to die, to, well, everything decreases at some point, so we'll be fine. And basically so I think that upper thermal constraints are not as universal as depicted in the literature right now. And most of the really good examples come from vector-borne diseases. So I think that upper thermal constraints as a strong limit on the spread of infectious disease is definitely something that we expect in vector-borne diseases, but I'm not sure how general it is, and it hasn't really been explored in other types of systems, at least not in a rigorous way at all. So speaking of sort of vector-borne diseases as a category, I think that disease systems where exposure is driven by the foraging rate of ectothermic hosts should be another cool category of disease. There's a baculovirus gypsy moth system that shows a similar pattern as the one we found here, where foraging rate increases with temperature, and so we see bigger epidemics as temperature increases. And so I think it would be cool to see if this exists in other systems as well. And then finally, I think trans-host effects could be really important, and they're basically not studied at all. I mean, they're studied very little in general, and they're not really studied at all in the context of temperature and seasonality. And I think I've showed here that they're really important for our system, and maybe they're also important in other systems. We just don't know because nobody's really looked. Well, they looked in one system and didn't find anything, so betting 500. Okay, so I'd like to thank, first of all, my advisor, Spencer Hall, who has been an amazing mentor over the past 6 years, and especially over the past month and a half, getting me out of here. He has put in way more work than he probably needed to or could justify to his family. Funding sources, Indiana University and the National Science Foundation. Spencer's main collaborators, Megan Duffy at University of Michigan, and Carla Castros at Illinois. Hall Lab members, basically all of whom are co-authors, and then other co-authors, Dave, a former grad student from our lab, Rachel, a former grad student from Meg's lab, Ali Magnanti and Andrew Sickbert, two really amazing undergrad researchers, and Daniela Vergara, who is a former grad student from the Lively lab here. Also NPR, and George R. Martin for keeping me sane during literally hundreds of hours of microscope work. And I want to specifically thank some grad students who are co-authors and were really instrumental. Both Dave and Rachel trained me and invested a lot of time and effort when I first got here. They both recently increased their evolutionary fitness and scored faculty jobs. Dave will be starting at Emory, and Rachel will be starting at WashU. Alex and Jessica, who are both really into birds, and have kept me company during the past 5 to 6 years, and we've collaborated a ton on basically everything till insane hours in the morning, and used co-authorship as a bribe on numerous occasions. And then the newbies in the lab, Jason and Maya, who are party animals, clearly, and just so fun to be around, and who are also co-authors on 2 of my papers. And then to everyone else who has made the past 6 years in Bloomington just a really great experience. And so,
questions for Marta? The confidence intervals on your transmission rate are sort of fan-shaped. So as it gets higher, they get broader. And they also seem a little asymmetric in the ones measured in D.
yeah.
Does that mean that the risk of super high epidemics goes up as transmission, as temperature goes up?
So I think, so in terms of the fan shape, part of that is just that you really can't get big epidemics when it's cold, but when it's hot, you have the potential for big epidemics, but there's so many other factors at play. Things like diluter hosts in the environment that are consuming spores but not becoming infected, the amount of dissolved organic compounds in the lake water can shield spores from solar radiation damage or not. And so basically, I think hot temperatures are sort of necessary but not sufficient for big epidemics, and that's one of the reasons why it's fan-shaped. And the reason why they're asymmetric, I think part of that has to do with the mathematics of, or the statistics for how we fit the transmission rate parameter, and just the way that the binomial distribution works. They're always asymmetrical with bigger on top. So I sort of, it's just sort of like an inherent part of a fitting a binomial process. Where, so yeah, basically like, when we do the statistics, we have hosts and we say, what's the most likely transmission rate given that this many became infected or did not become infected?
What's the temperature range they experience as they vertically migrate?
Ooh, that's a really good question. It depends a lot on lake habitat structure. So, So
the reason I'm asking is,
yeah,
in the immense cosmic experiments, you keep temperature constant.
yeah,
In real life, that
right.
is true. And I don't know anything about this context, but I know from rearing insects that there's a big difference between keeping them at 25 solid, or keeping them at a mean of 25 with a fluctuation around them.
Yeah, so there's a lot of research. It's like a very hot area right now, sort of how variation affects temperature. And we see different effects of variation depending on whether you're on the top side of the curve or on the bottom side of the curve. So if you're on the decreasing portion of the curve, variation tends to decrease trait means because you sort of get penalized for being in that zero area more than you might guess. And then on the other side of the peak, it actually tends to increase compared to what you would expect at constant temperatures. So that's definitely an issue. My experiment was not set up to look at variation per se, right? It was set up to sort of answer this very specific natural history question. So it's a really interesting question. I actually proposed it in my proposal and just kind of never got around to it. So if someone else wants to try and answer that question in Daphnia, I mean, I think both just for disease and also for like host traits in general. Actually, I was at a meeting in March and they were talking about temperature variation and how it affects traits. And one of the examples that they had was offspring reproduction in Daphnia. So I think there's a lot of research that could be done there.
Great talk. I really like the past temperature. That was really cool. And so it brings me to a question. What do you think is going to be more important for epidemic size? Like the temperature at which a lake gets to, or how long before mixing will evolve?
That's a really good question. So I am not well versed in physical limnology. Spencer is more well versed, and Spencer's collaborator at Ohio State, whose name I'm forgetting right now, is even more wonky about that. And so it's something that we're looking into, and we don't have a good answer for how mixing. I mean, we know mixing is important, and we know that a lot of times, like you can look in a specific year. For instance, I think it was like 2010. It was like really hot, a prolonged hot period, and then a big storm came through, and all of the lakes mixed. And then literally the next week, epidemics started in every single lake. And
Marta, you have something to say about from your simulations about when cooling happens.
Oh yes.
Good answer.
So let me see if I can.
You just didn't show it.
Yeah, so this was in the chapter that I submitted to my committee, but I just for time didn't include it here. That so here we have sort of one temperature profile, and then we vary the traits. I did a whole nother set of simulations where we have all of the traits vary with temperature, and then we vary temperature profiles. And so we can show that if mixing happens later, then you'll get smaller epidemics. Or sorry, if cooling happens later, then or sorry, epidemics will be larger. Yeah. So the more time they have to go at these hot temperatures, the bigger epidemics will be. We can also vary starting temperature, which relates to variation in physical structure that we see in nature. We have a lot of lakes that are sort of like 15 meters deep and a lot of lakes that are like 8 to 9 meters deep. And so the shallower lakes get much hotter in the summer than the deeper lakes do. And a lot of times we see bigger epidemics in those lakes as well. Thank you.
I think this sort of relates to Maria's question. So I'm in Daphnia in August.
Yeah.
What spores, where are they from?
That's a really good question and something that we've looked at. So the hosts are definitely there before epidemics start. And so we don't think that it's hard to know when the spores are getting mixed up, but we think they're basically coming from the bottom, and through some sort of physical mixing process, they get brought up into the layer where the hosts are hanging out. We did an experiment, I think this past spring or maybe one spring ago. Time is hard to figure out at this point, but basically, so mixing happens twice a year. It happens in the spring and it happens in the fall. But in the spring, our hosts aren't there. They don't show up until the summer. And so we went out into the lakes right after they mixed in the spring, and we collected water. We brought it back to lab and we fed it to our host to see if there were spores getting mixed up at that point, and we didn't get any infections. So I don't really know what to say about that. I mean, you would think based on sort of like our conceptual model for how epidemics get started in the fall that they would also get mixed up in the spring, but like I was saying before, like I don't know that much about physical limnology, and it seems kind of complicated. Mike?
Whatever natural selection goes on in the Daphnia, if the infection prevalence is 20%, selection is 2.5 times stronger, prevalence is up 50%. So how much variation is there between Daphnia clones and the features in the
So there's a lot of variation in Daphnia. There's basically no genetic variation for infection in the fungus, but a ton in the Daphnia. And I would say that like several months ago, I would have been able to answer your question, but after listening to Jason and Alex talk in the lab about some results that they have right now, evolution in response to infection seems kind of complicated, and sometimes you can get really counterintuitive results because so infection is really driven by exposure, which is really driven by foraging rate. But if you have a high foraging rate, you also eat more food and can make more babies. So there's the trade-off between fecundity and susceptibility. And we actually see evolution of susceptibility in a lot of situations in which you might not expect it. And so I can't really talk about why or how, but if you wait a year, I think Alex will probably be talking about that, or no, half a year in December. Alex is gonna have a lot of really interesting things to say about host evolution. Alex?
Probably an A question about how hot this is. What, I guess,
293 Kelvin.
also, my question is like, in what situation can this interaction get onto 31 temperature?
So the shallow lakes quite often reach like 32, 33. The deeper lakes tend to only get up to like 27, 28. Because there's just so much more water they have to go.
Is that your weighted temperature, your aluminium?
So there is talking about epilumnetic temperature, so that's the surface layer. The weighted temperatures are more like 26, 27 in the shallow lakes and 23, 24 in the deeper lakes.
So on that, did you have a method for pulling Daphnia from
lower depths, or did you only pull them from the surface?
So when we collect Daphnia, the way we do it is we take a vertical sample. So we have a net and we sort of like throw it off the side of the boat, put it down until it hits the bottom, and then we pull it up. So we're sampling the whole vertical spectrum. So there are methods that you can use to take samples at specific depths, but I don't have any data from that here.
And are you getting multiple different species of Daphnia?
Yeah, so again, if you come to Alex's dissertation, he'll have a lot to say about host community composition. There's 3 main players, so including the focal host that I was talking about today, and then 2 others. And then there's a bunch of other randos that just show up sometimes.
All right, let's thank Marta.